Hi, my name is Casey Sands, and this summer I research at Rockefeller University in the Strickland Lab of Neurobiology and Genetics with my mentor, Dr. Karen Carlson. So we examine the extracellular matrix protein laminin in mouse blood vessels of the liver and tumor in order to view its effects on vascularization. First, a little background on laminin. It's an extracellular matrix protein, generally functioning as a structural support for cells to develop and grow on all throughout the body. One very important use of laminin is the formation of the basal lamina. The basal lamina, or basement membrane, is a membrane surrounding blood vessels and is essential to the development and health of blood vessels. Its main functions are being a structural support and monitoring what enters the blood vessel, also cell differentiation. So in my research project, I was looking at the effects of laminin produced by a particular type of cell on blood vessels called the pericyte. Pericytes are mesenchymal-derived cells that surround endothelial cells. So endothelial cells are cells that surround and support the blood vessel. This is my diagram of a blood vessel, including the pericytes, endothelial cells, and basal lamina. As we said earlier, laminin is key in supporting a healthy blood vessel. So we wanted to know where the laminin was coming from. There's evidence to believe that endothelial cells do not make enough laminin to support themselves. So the extra laminin would have to be produced by another cell nearby the blood vessel, like the pericyte. The reason the source of laminin is so significant is because it affects the growth of tumors. And so a little close up on here's so tumors, masses of cancerous cells need oxygen via blood flow to survive and grow rapidly. Therefore, the knowledge of where an important protein like laminin, which isn't needed for blood vessel formation, originates, is valuable in finding ways to combat the growth of cancerous tumors. All of the mice in this experiment were injected with a known strain of Lewis lung carcinoma cells so that we could study parasitic laminin's role in cancer. To target the laminin produced specifically by pericytes and not endothelial cells, we used an enzyme transgene called pre-recombinase with a nestin promoter to cut out the laminin-making gene in cells that produce the filament nestin, a filament expressed by pericytes but not endothelium. And that's my little animation of that one. So we looked at tumors because we wanted to examine the importance of parasitic laminin on the blood flow to tumors, and the liver because the liver has special small blood vessels called sinusoids, vessels that have an incomplete basal lamina, which allows cells to move in and out of the bloodstream more easily. On the screen is a picture of a sinusoidal vessel. These vessels are the easiest access for cancerous cells to enter the bloodstream and travel throughout the body. After examining the livers and tumors of both mutant and control mice, we found some interesting results. Firstly, mutant mice tumors had fewer small blood vessels than the tumors of control mice. As you can see on the screen, there are two stained images of tumors, a control tumor on the left, and a mutant tumor on the other side. Red are blood vessels, green is laminin, and blue is nuclei. As you can see, the amount of laminin in the mutant mice is significantly smaller than that of the control mice, and the number of blood vessels of the mutant tumor is lesser than that of the control. This finding is supported by my next result, increased necrosis in the tumors of mutant mice. Necrosis is cell death, in this case caused by lack of oxygen to the dead tissue, meaning that there is an insufficient blood flow to the mutant tumor. This is because of the decreased number of blood vessels in mutant tumors as seen on the previous slide. Dead tumor cells will not divide and therefore slow tumor growth. Dissimilarly, the control tumor is sustained and growing. Now looking in the liver. The hepatic cells, or liver cells, of the mutant mice showed significantly less laminin than that of the control mice, to a greater extent than expected. The blood vessels of the mutant liver were also dilated, far larger than the vessels of the control, signifying abnormal growth of these vessels due to parasitic laminin. The dilation of the hepatic sinusoid vessels is demonstrated by the yellow lines measuring the length of the vessels. Lastly, we did not see metastasis, the spread of cancer, in either control or mutant mice to the liver or lungs. However, this could be because of the length of time the mice's cancers were allowed to survive and spread. That leads me into possible future research. For example, I believe that parasitic laminin may play an important role in the metastasis of cancer. Finally, I would like to thank Karen Carlson, Zulin Chen, Puni Kermani, Sydney Strickland, Ted Scoville, all of Rockefeller University and the Hematology Department at Cornell, and Dr. Cox for putting this all together. Thank you.